So today we will be talking about the catecholamines and its synthesis, then the action termination by reuptake and metabolism, and these all will be under topic adrenergic, or more precisely non-adrenergic transmission. And from this synthesis, this pathway, you will be frequently asked write down the synthesis and degradation of the catecholamines, or there is one more transmission, that is parasympathetic transmission, wherein acetylcholine, synthesis of acetylcholine and its degradation or action termination will be asked in your uni or internal exams. So these two pathways are very important questions in biochemistry ANS block. And from this pathway, some question may also be asked in pharmacology, mainly MCQs. Then I will be showing that MCQs too. And along with this pathway, we will be also dealing with the clinical significance and relation with the medicine, that is pharmacology. So let's start. Catecholamines. Catecholamines. What are these? The examples will be dopamine. It acts as neurotransmitter mainly in central nervous system and more precisely in basal ganglia or basal nuclei and some other parts of the brain. Then here will be noradrenaline. This is the main neurotransmitter at post-junctional sympathetic fiber. You know already, I have discussed about this in the physiology, ANS physiology, and there will be adrenaline. This adrenaline is synthesized and released by the adrenal medullary cells, and when sympathetic nervous system gets activated, these adrenal cells also gets stimulated and releases both adrenaline and noradrenaline and mimic the same action as like sympathetic actions done by noradrenaline. So you know this very well, and you should know. Sometimes in your questions will be asked like, describe the pathway synthesizing catecholamines and its degradation. So how the synthesis occurs, let's see. This is nothing but benzene ring. You must know it from the knowledge of the chemistry till your +2 level. CH2CH and H2 and H2 and COOH. That is, this is your amine group and this is carboxyl group. If in any molecules, biomolecules, there is two groups, amine and carboxyl group, you know this will be called as amino acid. Amino acid. So this is nothing but amino acid. Phenylalanine. This is phenylalanine. Now this phenylalanine will undergo catalysis and this phenylalanine will be converted into another amino acid, CH2CH, COOH and NH2. Here hydroxylation reaction will occur and at para position hydroxyl group will be added. So change is here. What will be this converting enzyme? This is phenylalanine hydroxylase. What is this? This is tyrosine. Now this tyrosine will be converted into again by hydroxylation reaction at, I think it is meta position. Confirming your knowledge of chemistry, you know it well. Here one more hydroxylation will occur, and this part will be the same. CH2CH and H2 COOH. And this will be LIVO, L for LIVO, rotatory form and dextro rotatory form. You know, in enantiomerism there will be one form called LIVO rotatory and another form called dextro rotatory. So this is LIVO rotatory and DOPA. This is DOPA, LIVO DOPA, and you know this by knowing this LIVO DOPA, in case if there is deficiency of the catecholamines, mainly the dopamine, there is LIVO DOPA is given for that patient. Tyrosine, this will be LIVO DOPA, and the reaction will be carried out by the enzyme tyrosine hydroxylase. Tyrosine hydroxylase. Tyrosine hydroxylase. From this LIVO DOPA, I will writing from here, from this top. LIVO DOPA. From LIVO DOPA, it will be converted into dopamine. dopa dopamine, which here CH2 and H2. Now it is only the amine. Here is only amine group. This is amine. We call it as dopamine. Dopamine. The reaction will be carried out by enzyme. Here is decarboxylation reaction occurring. Decarboxylation. ADC, or you can call it as aromatic amino acid. Aromatic, again put here L, levo, amino acid decarboxylase. Or simply you call it as dopa decarboxylase. Dopa decarboxylase. Dopa decarboxylase. At least remember this name. So from this pathway you are seeing that these amino acids are these two, phenylalanine and tyrosine, are the aromatic amino acids. In general biochemistry, besides essential and non-essential amino acids, you may also be asked in viva or MCQs enlist or enumerate the names of aromatic amino acids or amino acids containing sulfur. So what will be the aromatic amino acids? Here is a two example, phenylalanine and tyrosine, and there will be one more aromatic amine, that is tryptophan. So remember the name. And from this pathway what you can also notice that this tyrosine is forming from the amine phenylalanine. What does it mean? If in case tyrosine is not consumed, it will be formed from the phenylalanine. That is, this is non-essential amino acid. Why? Because this amino acid can be synthesized in the body, and your definition is also same, that amino acids that is synthesized in the body from any other precursor or any other amino acids, that will be your non-essential amino acids, and that what could be included in your diet or that cannot be synthesized by body, that you are calling essential amino acid. And here is the example of one essential amino acid, that is phenylalanine. So remember, everywhere you can find the your knowing point, what you should know or what you need to know to tackle exams, theory, MCQ or viva. So I will read dopamine. Now this dopamine will be converted into noradrenaline. CHOH CH2 and H2. This will be noradrenaline. Benzene ring, here will be the hydroxyl group, and here also hydroxyl group. noradrenaline. The enzyme converting this will be dopamine beta hydroxylase. Again here is hydroxylation reaction at beta position. And you know, from any functional group, if you number the other carbon atoms besides 1 2 3, you can also number it as alpha and beta. You know the chemistry very well. At beta position here hydroxylation is occurring, so enzyme will be dopamine hydroxylase. Now finally we done two preparation. Here adrenaline is left. So adrenaline, how it is formed? CHOH, CH2, CH3, sorry, here will be N, CH3 and hydrogen. What is occurring here? Here methyl group is added, so the reaction will be methylation, and the enzyme will be phenyl ethanol amine in methyl transferase. At least remember N-methyl transferase. So here methyl group is added, and this will be your adrenaline. And sometimes you should know some locations where these reactions are occurring. If not all locations, at least you should remember that this adrenaline is found in the adrenal medullary cells only, not other sympathetic or other nerve fibers. So location. This first reaction will be occurring in liver. Location will be at liver, and these two reaction will occur in adrenergic neuronal cytoplasm. What is it? I will show there. This reaction and these two reaction will be occurring in adrenergic neuronal cytoplasm, and this reaction will be occurring at adrenergic vesicle, inside adrenergic vesicles. Location, and this very important location at adrenal medullary cells. There is adrenal gland over the upper pole of kidney. You also call it as suprarenal gland, and there in suprarenal gland there is two part, one cortical part and one medullary part. Cortical part will synthesis and release the corticoids like glucocorticoids, mineralocorticoids and sex corticoids, and these medullary cells will synthesize and release adrenaline and noradrenaline. So this reaction will occur in adrenal medullary cells. At least remember this. And here also I would like to write some cofactors associated with this reaction. For example, this reaction, for this enzyme, the cofactor will be S-adenosyl methionine. I am writing a short form, S-adenosyl methionine. And then first reaction, the cofactor will be tetrahydroxybiopterin. Tetrahydroxybiopterin, and this reaction will be oxygen dependent. This will be your cofactor. Here also cofactor cofactor, yes, and here will be the cofactor vitamin C, cupric ion and oxygen. And this reaction here will be the cofactors PLP, pyridoxal phosphate. It is the active form of vitamin B6. In vitamins, we will be asked MCQs of any vitamins or active forms of any vitamins. For example, for vitamin B6, the active form is pyridoxal phosphate. And this so just what I want to explain is that cofactor, you must have been taught that most often the vitamins act as cofactor. And you are seeing here this pyridoxal phosphate, vitamin C, and some other, and that is this reaction is oxygen dependent. So oxygen is also needed here, and other ions, that is cupric ions. So the purpose of this knowing is that if there is deficiency in any of the cofactor or enzyme, what clinical significance can occur, you can notify. So this is the synthesis of the catecholamines. Now the important point, from any biochemical pathway, what you are supposed to learn or you should learn is that if there is any defect in the pathway, or precursor, or if there is increased synthesis, the intermolecular synthesis, biomolecular synthesis, if there is increased, how we can decrease it, or if there is any enzyme deficiency, what clinical significance can occur. And also here and degradation, we will be seeing the clinical relation, that pharmacological relation, how if there is these molecules are deficient or reduced, how it can be prevented from the degradation and the action of these molecules are maintained. These all are pharmacology. So here what I am trying to say is that this this tyrosine hydroxylase is rate limiting step, rate limiting enzyme, or this step is rate limiting step, rate limiting step. That is, if here is any defect in this enzyme or this pathway is not proceeded forward, deficiency of all catecholamines will occur, and this enzyme is blocked by alpha methyl p tyrosine, alpha methyl p tyrosine, or simply you can call it as metirosine. This metirosine is important enzyme, drug that will be inhibiting this tyrosine hydroxylase enzyme and the pathway. And why we should know this? Because in some cases there is excess production of the catecholamines and the actions will be exaggerated. So to reduce this, if you know this pathway, we can stop or reduce the pathway here and its synthesis. So this is important point. And this drug is used in case of pheochromocytoma. In case of pheochromocytoma. What is this? This is nothing but abnormal proliferations of adrenal medullary cells, and there will be increased production of adrenaline and noradrenaline, and the actions of these molecules will be exaggerated. So this is the exaggerated action is not needed. So in this pheochromocytoma, this drug is effective. So how we apply the drug in pheochromocytoma? By knowing this pathway. So this will be your pathway synthesis of the catecholamines. And for exam point of view, remember, if you don't know the structure, no need to write, and if you also do not know the sites where the reactions occurring, no problem. If you know, that is better, no problem. What you should at least remember is that phenylalanine converted to tyrosine by enzyme phenylalanine hydroxylase, or at least you remember hydroxylase. First two reaction is hydroxylation reaction. Hydroxylase. Here dopamine, levodopa, from levodopa, this enzyme AADC or dopa decarboxylase. At least remember dopa decarboxylase. This dopamine, if you don't know the structure, write dopamine beta hydroxylase. Here again, at least remember beta hydroxylase. Here is also hydroxylation reaction occurring at beta position, and this will produce noradrenaline, and at adrenal medullary cells, adrenaline will be produced from this noradrenaline by enzyme phenylethanolamine N-methyl transferase. At least remember N-methyl transferase. And regarding these cofactors, some cofactors, this is correct and this is correct. There are two cofactors. I am confused. PLP is used here or here. So cofactor what I only want to explain in cofactor is that this cofactor most often are vitamins, or if there is any deficiency in that what clinical significance can occur. So this is our synthesis pathway. Now we will be moving on to the its degradation or metabolism, or how the action is terminated. Action termination of catecholamines. Catecholamines. So here there is multiple pathway for terminating the actions of catecholamines, and we will see the major and minor pathway. As soon as this catecholamines is released from the axon terminals, after the action is desired action is performed, the action should be terminated. So for action termination, here is important pathway: reuptake of catecholamines from synaptic cleft. This will be occur in 3 steps. Main step is axonal uptake. This axonal uptake, let's suppose this is central nervous system part or spinal cord. Here is the axon coming. This will be adrenergic vesicles or axon terminals, and it will release the catecholamines. Let's suppose noradrenaline. So after release and desired action is performed, the action should be terminated. How this occurs? Axonal uptake. This is synaptic cleft. Let's suppose synaptic cleft, and this is postsynaptic membrane. So here is action performed on postsynaptic membrane, and desired action is performed. This noradrenaline should be taken back. The major pathway is axonal reuptake. And this axonal reuptake will contribute to 75 to 90%. That is maximum of the catecholamines is taken back by axonal uptake, and the action is terminated. That's the major action termination pathway will be axonal uptake of catecholamines 90%. And this is your very important MCQ question. Keep it in not in gold but in platinum category. This MCQ in platinum category, MCQ and Viva. How the question will be asked? Question: the major mechanism of action termination of catecholamines. Let's suppose here put noradrenaline. Action termination of noradrenaline is by which mechanism? Your answer will be axonal uptake. Axonal uptake. This is very important MCQ question, and some other options will be also there, which is not correct. Why I am focusing on MCQs? Because you can see in KU pattern there is 30 marks MCQ and 50 marks SAQs, and MCQs will not let you at any competitive exams like in license exam, or if you try after graduation, if you try to go foreign like US, MLE, or some other examples, competitive exams is mainly by the MCQ pattern. So if in case I think in TU there is no MCQ, there is no provision of asking MCQ in the undergraduate level. So you should also, if you remember the MCQs, it will be helpful later. And regarding MCQs, remember one line: the MCQs can be made from the any line or every line of the book. What I said, the MCQ can be made from the every line of the book. Means if you know the theory well, it is not big deal for you. MCQ is not big deal for you. But there are some important points that must be or frequently asked in only as MCQ at your level or higher level. So I will be mentioning that MCQ point also, and this will be the distinguishing feature of the FA medical lectures, that along with SAQs and concept of theory, I will be also talking about important MCQs and some frequently asked Viva questions also, so that from this lecture, from this channel, you will be getting the enough answers that will be asked in your exams. So here answer is the axonal uptake. And some other minor pathway will also be there, like vesicular uptake. Vesicular uptake, what is this? If this is at first, this noradrenaline is will be transported into this adrenergic cytoplasm, or noradrenergic cytoplasm, and from this cytoplasm it should be transported to the inside vesicles. So this vesicular uptake is that taking the noradrenaline back to the vesicles, and it will be reused. And some other pathway also there, like some other tissues will also uptake this noradrenaline in few amount and the help in the action termination. So this is the major pathway of action termination. And this, remember, while I will be dealing synthesis of the acetylcholine, I will mention there also the, and there is also very important MCQ question, the action termination. The action termination of acetylcholine is by which mechanism? The action termination by action termination of acetylcholine. This action termination will be done by the degradation by enzyme acetylcholine esterase into choline and acetate. So here MCQ is the major pathway of the action termination of the parasympathetic nervous system or acetylcholine is by degradation by the enzyme acetylcholine esterase. And here also again I will add the degradation by the metabolism. It's metabolism of the catecholamines. By metabolism, some catecholamines will be degraded and the action is terminated. Metabolism, and metabolism I will explain in detail. Metabolism of catecholamines. How this occurs? Let's suppose it is epinephrine. Adrenaline you also call as epinephrine, and noradrenaline as norepinephrine. This will be converted into metanephrine, and here normetanephrine, and finally this normetanephrine and metanephrine will be converted into 3-methoxy 4-hydroxy mandelic acid, or you call it as vanillyl mandelic acid, that is VMA. And what will be the enzyme here? Degrading enzymes. Here will be your COMT. What is this? Catechol-O-methyl-transferase. Here also will be catechol-O-methyl-transferase, and at next step here will be monoamine oxidase, and the final end product will be the vanillyl mandelic acid. This is also MCQ question, that what is the degradation or end product of this epinephrine and norepinephrine or catecholamines. And if this reaction, this enzyme is directly act first, if monoamine oxidase is act first on norepinephrine, monoamine oxidase, and here monoamine oxidase. Some other intermediates will be found, and final end product will also be the same. Intermediates, other intermediates will be found, and again this will be converted into the by catechol-methyl transferase will be converted into the mandelic acid. So this is your degradation pathway. Now, what I earlier said that from any biochemical pathway, you should know the clinical relation, that if there is excess production, then how can we reduce that production, or if there is excess degradation or deficiency, less production, how we can preserve it and its action will be preserved. This is the biochemical pathway. By knowing this biochemical pathway, then later you will be studying, you will be learning anti-Parkinsonian drugs. Why these drugs is used? Because there is deficiency of the dopamine, and Parkinsonism is occurring. So, and by this pathway, by these enzymes, dopamines will also be degraded in same end product. Then here COMT inhibitor. This inhibitor is used like entacapone, tolcapone, and if this enzyme is inhibited, the degradation will be prevented or reduced, and the dopamines will be preserved, and the action will be maintained. Here also, and other examples, other drugs is used in anti-Parkinson drug is that monoamine oxidase B inhibitors like Gilin and its congener. So if you know this pathway and the enzyme, you can either potentiate the action of enzyme or you can inhibit its action. And what the desired action is you can maintain this by means of the drugs. So any biochemical pathway, what I said, in biochemical pathway, how drug we can apply, or if there is any defect in the pathway, what clinical significance can occur, we will be learning from this, or any biochemical pathways. So there are many other drugs also used in anti-Parkinsonian drugs. You will be studying in your second year in central nervous system block. So these were our synthesis of the catecholamines and its action termination or degradation. And if you ask any pathway, the synthetic, if you asked in exams in long questions, and also if you have time in synthetic pathway, besides writing the synthesis, you can also add on its degradation, so it will give the sense of complete answer if you know the degradation also. Otherwise, at least you should write the synthesis what is asked. Same as like in anatomy exams, after writing all answers, if you add on the applied anatomy or applied aspect, your answer will be giving complete sense of answering, and you will be getting either full or close to full mark if you write the all required thing. So hope you enjoyed the lecture. Thank you.